start this session with Craig Kundrat, who is the Division Director for Biological and Physical Sciences at NASA headquarters. And then we will have a series of 3 talks, then 20 minutes Q&A, and then 3 early career talks. So welcome, thank you again, and Craig.
Thank you, Sharmila, and I echo Sharmila's thanks for being here at 8 o'clock on the last day, on a Saturday. Looking forward to tonight where we all get an extra hour of sleep. I just have a few remarks. It's been an evolving picture sort of month by month with our move to the Science Mission Directorate. And it's a very exciting change for where we have been in the last 10 years. In the Science Mission Directorate, there is a joy and delight in science and an aspiration to seek out and do the most compelling science. And happily, it's instantly recognized that in space biology, the effort to thrive in deep space stimulates compelling science of 2 types. First is the basic scientific knowledge, and then second is laying the foundation for human exploration to do sustainable exploration beyond low Earth orbit to the moon and on to Mars, and eventually who knows where. So it's really an enjoyable change, and it's great to have that excitement and focus on compelling science. And what you'll hear about from Sharmila is our near-term plans in space biology, but the long-term plans are going to be shaped very, very heavily by the decadal survey that's underway right now. And I just want to emphasize the importance of that activity. It's community-led, and a major part of that process is the white paper process. And we can't stress its importance enough to you. So white papers are 2 to 5 pages long. Right now, they're still taking white papers until December 23rd on what are called research campaigns. There is not an official definition of what a research campaign is. It stands in contrast to a topic area. So it could be several lines of investigation that's answering an overarching question. It could be a sequence of platforms and destinations. There are a lot of possibilities. But I strongly encourage you to share all your exciting ideas. There's no penalty for an idea they don't accept or something that doesn't fit in the definition of research campaign. It's better to err on the side of inclusion, because the decadal survey will be shaping the face of space biology for the next decade and laying the foundation thereafter. So in the meantime, as Sharmila is going to share with you, we are very excited about the emphasis on thriving in deep space, and Sharmila is about to tell you what that means. So over to Sharmila. Thank you.
Thank you. Thank you very much, Craig. So there we go. I'm going to test this out. There we go. So for the agenda today then, I will start with an overview of the program. I'll talk about and describe when we say thriving in deep space, and we call it TIDES, what exactly we're referring to. We'll talk about the solicitation activities, and then 6 transformative science areas that the larger science community uses right now that we think could have a lot of potential benefit for us in space biology. So space biology, as many of you may know already, and for those of you who are new to the community, and we welcome new folk as well, is we're really interested in understanding the changes that are undergone by Earth-based biological systems when they're put in space. We are NASA's fundamental research program for biology, so we're really interested in understanding mechanistic changes and then using these to, for example, understand, to predict and estimate what might happen to humans, as well as utilize systems like plants, which we know will be important for long-term habitation in these deep space environments. To introduce you to the team, because several of the faces may be new to the community, I wanted to introduce Mary Walsh, who's in the audience here. She's the program manager for space biology. She's on detail from Johnson Space Center. There's Mamta Nagaraja, who's also in the audience, sitting in front here, and she will help us moderate the early career talks in the second half of this program. Anthony Hickey, many of you will know, has been with space biology for some time. Kevin Sato, I'm sure all of you know as well. Kevin is the program scientist for exploration, and he will be speaking right after me about deep space exploration opportunities and plans. Lisa Carnell, who's the program scientist for translational research. So she works closely with and helps liaison with NASA's Human Research Program, which is a more applied program looking at risks of spaceflight to human health. And she also works in partnerships with other governmental agencies. And Doug Grandel, who's our partnership coordinator. So then the way I wanted to start is really drill down into what are some of these stressors that we will encounter as we go into space, low Earth orbit and then beyond low Earth orbit. So the first one that I wanted to point out here is the microgravity reduced gravity. Now, for Earth-based living systems, when, over a long period of time, the systems have evolved in a 1G environment. So when you are either on the International Space Station or when you're on your journey to the moon or to Mars, the body will experience microgravity. And then reduced gravity on the surface of the moon, for example, one would encounter about 1/6 the gravitational force that you would on the surface of Earth. So again, for Earth-based systems, this could be a challenge, and we know from data from the International Space Station, and many of the presentations you heard at this session points to the fact that there are changes that result from changes in gravity. The second one I wanted to talk about is ionizing radiation. So as we, on the surface of the Earth, we're really protected by Earth's magnetic sphere, by the Van Allen belts. But when we go beyond that, we will experience larger doses or larger levels of this ionizing radiation that will impact biological systems. And there's two flavors of this ionizing radiation that we would encounter. One, which is really damaging to biological systems, are the galactic cosmic radiation. This consists of charged particles like iron, oxygen, silica, and so on and so forth, which are present there in low dose and low dose rate, but they're there continuously and they're very hard to shield against. And when they come in contact with cells or tissue or biological systems, they really can, as they blast through the cells and the tissues, they really can cause a lot of damage. So that is one that we really want to make sure we understand the effects of. The other complexion of this radiation are protons coming from the sun. These are easier to shield against, unlike the galactic cosmic radiation particles, but they're stochastic in nature. So unlike the galactic cosmic radiation that have a continuous presence and a predictable presence, the protons from the sun come in bursts, and you can get a large dose and you don't know exactly when to expect it. But again, they can be shielded against. So that's the ionizing radiation component. Then you have the fact that when you're in space, essentially, and in fact, when you're on the lunar surface, for example, when you compare lunar day to lunar night, there can be an almost 300 Celsius swing or difference in temperature between seeing the sun and not seeing the sun. And the atmosphere also is inhospitable for Earth-based living systems. And so to protect ourselves, we engineer habitats then that can keep us protected from some of these factors. But as a result of these engineered habitats, though, we do have, for example, we would encounter elevated carbon dioxide levels. So there's almost 10 times more concentration of CO2 on the International Space Station than you have on the surface of the Earth, even though CO2 is scrubbed actively. But again, it's this enclosed environment, and it brings with it certain changes. Also, when we want to make sure that astronauts can do EVAs safely, there is, for example, when we're on the Gateway or on the surface of the Moon, on habitats there's the chance, the likelihood that there'll be reduced atmospheric pressure and elevated volumetric fractions of oxygen. Other stressors that we would encounter would include altered day-night cycle depending on the orbit that you're in. That one can counter with artificial lights in your habitat, for example. There's also isolation. Just the mere fact that you're months away when you're on the surface of Mars, for example, months away from being coming back to Earth. So if you need something, in an emergency or for medical reasons or even for your experiments, it really becomes very difficult to go back and forth. And then not to mention we have constraints on up mass and return mass and all of that. So really it behooves us to be as self-reliant and independent when we are on those surfaces, especially when we're in Mars. And so the research that we do here in space biology really aims at that. We are trying to understand what some of these changes will be, anticipate them, and then know what it is we need to mitigate against it, that we can carry things with us or take whatever precautions we need to do. And lastly, I wanted to mention regolith, and I'll come back and talk about that a little bit later as well. Regolith is something that we will encounter on the surface of Moon, and it's something we could use to our benefit. So, for example, we might be able to use it effectively for growing plants, but more work needs to be done in that area to ensure that we can do that effectively. And then on the other hand, also, there are potential health impacts to astronauts when they come in contact with the material. And so these are all, showing you all of the stressors on this slide to give you that sense of there are multiple things that we will be encountering. And while on Earth we might have studied things in onesies and twosies, really, when we go into this environment, it's going to become important for us to understand the holistic picture of what is happening. So this concept, as Craig mentioned, of thriving in deep space, and it's really one we've embraced because we feel it's very important for our journey and for NASA's mission of getting us safely back for sustained presence on the Moon and then onto Mars. And two of the stressors, I discussed the stressors a minute ago. The radiation and gravity stressors I wanted to stress on to really point out that while to some extent we can simulate these effects on the ground, and we do, and they are effective in helping us prepare for spaceflight missions, but these are two stressors that are very hard to accurately represent or replicate on the ground. And so it really becomes important for us to do those spaceflight missions to understand the full impact of those on biological systems. And then in terms of the systems that we're interested in using to do this research and understand these phenomena as we described it, animal biology, of course, model systems, vertebrates and invertebrate systems, to really begin to understand and predict the changes that might occur, for example, in humans. Similarly, for plant biology, we're very interested in understanding the changes on model plants and learning how we can really robustly grow these plants in the space environment. And then in terms of microbiology, as we all know, microbes can either help us and can help the animals and the plants thrive in deep space. And then there's a category of microbes that might be detrimental to the health of animals and plants. And we need to understand those interactions as well, because we will inevitably be carrying or taking microbial species up with us. So to summarize this idea that you may hear from us periodically over the next several years, is this concept of thriving in deep space. And to summarize that then, on the left side, the building blocks here are, one, on the left side, understanding the fundamental mechanisms. Again, we are the basic research arm of NASA, and we're really interested in understanding the science that goes behind the changes that we see in space. The middle block refers to plants and really understanding, again, how we can sustain plant life that will help sustain us for long-duration missions in deep space in a self-reliant manner. And then all together, this research will then help us integrate this idea and the stabilization of this human animal microbial ecosystem, stabilizing this, plant, human, animal microbial ecosystem is, and seeing how these will respond with these multiple stressors will help us build that research program that will allow us to be in this environment for periods of time. I covered the deep space stressors on an earlier slide, and then the platforms. The platforms that we can use to do this research encompass, of course, ground studies will continue to be important as we use them for preliminary studies to really start to understand what's important for us to look at out there. Suborbital vehicles, as well as low Earth orbit platforms, the International Space Station, and then whatever comes next, commercial low Earth orbit stations, for example, will be another workhorse for us to do our experiments in low Earth orbit. And then as things come online, and you will hear Kevin talk more about this, as we get the Gateway online, and as we start building habitats on the lunar surface or capabilities on the lunar surface for us to do science, that's the goal for us then to start looking at some of that in preparation for going to Mars. And then lastly, in terms of the organisms, the model organisms that we use in space biology, the good thing is we're at liberty to really test any model organism that we choose that best fits the science question that we want to answer. And so, for example, we can use a whole panel of organisms, including unicellular simple systems, for some of those early studies in deep space that are hard, to get resources for in terms of, getting any crew time help and so on. And then we can also use in the animal and the plant arena, we can go from simple to more complex organisms, invertebrates and vertebrate models, plants, simple model plants, and so on, and really holistically understand the effects of these environments using all these tools, all these platforms, so that, again, we can really get all of us safely in deep space for periods of time. Now for some of the recent solicitation activity, to update you on that. You might have seen that we recently announced the selections in early September for our last annual Roses Call. And just to let you know, as Craig mentioned, we have recently, about a little over a year ago, moved to the Science Mission Directorate. And the Science Mission Directorate uses, for their solicitation, uses the Roses system. It's not hugely different from what many of you may have been used to in the past, but there are differences. And so we now use the processes that SMD employs, and those are the Roses calls. So on NSPIRES, be sure to make sure that you have, you sign up to get notifications for Roses. And so with these announcements, we actually made awards to 10 investigators for a total of $9 million. You might also have seen that earlier this year we had a second call go out, which was for LEA, or these were Lunar Explorer instruments for space biology. And this was actually for a ground study to get ready to integrate into hardware so that we can do lunar studies. And we made 3 awards for this particular call, totaling 0.9 million. Congratulations, and I know this is an eye chart, and really it's not meant for reading really right now, but it is really by way of a reminder to congratulate and to welcome our new batch of PIs and co-Is and students and team members for the grants that were recently awarded in these 2 calls that I mentioned. And then there was yet another solicitation that we put out this year as well, a third one, regarding 3D tissue chip. And this was actually a partnership not only within NASA with space biology and the Human Research Program, but wider utilizing and partnering with NIH's various institutions, BARDA, and FDA as well. And this was released this summer, and the proposals were due in mid-September, and the selections will be announced early in the following year. And the topic of this was the extended longevity of 3D tissues and advanced microphysiological systems to model acute and chronic exposures to stressors. And like I mentioned, it's a partnership with a number of different governmental agencies, and the point of contact for this particular solicitation is Lisa Carnell, and as I mentioned, she works the other governmental agency partnerships in our group. So for upcoming Space Biology's Roses solicitation, we are anticipating putting out the next annual Roses call in the winter, so coming up in the next several months. And then the proposals will be due in the spring timeframe, and the selections will be announced in the summer to early fall timeframe. And we do anticipate putting out a second call as well, which will encompass, again, looking at experiments that we can do as we go into deep space. So, here again is our effort to both continue the research that we're doing on the ground as well as in low Earth orbit, and then also start venturing out into getting ourselves set up to do experiments beyond low Earth orbit. Other upcoming solicitations, we anticipate 2 more solicitations this year as well. One is the EPSCoR Rapid Research Response. These are typically 1-year studies for about $100,000. And the second one, which is the CAN, or the Cooperative Agreement Notice, also from EPSCoR, typically is a 3-year grant covering $750,000 per award. Now, just to orient you to the EPSCoR, in case you're not familiar with the EPSCoR opportunities, the EPSCoR opportunities target states or parts of the country that are underrepresented in terms of science and research for space flight and aerospace related science and technology development. So, so there are certain states where the EPSCoR grants, are very popular and make a lot of sense. Other upcoming activities, we also have yet another solicitation out, which is the top one listed there, which is it's a partnership actually with the NSF, with NASA, and with DoD. And just a heads up, the letters of intent for that are due on November 10th. and preliminary proposals are due on December 16th. And so you can go to that website to look at a little bit more of the details. And the POC for that is Lisa Carnell. And if you go to the website, you'll see the topic for this call is called Engineered Living Systems. And so it's looking again at systems. The NASA point of view there is that we're looking for engineered living systems that will help us, again, for sustained survival in deep space. So check it out if you're interested. In terms of workshops, I've listed 4 different workshops there. The Spaceflight for Everybody workshop is coming up soon, next week actually. The Gene Lab Analysis Working Group virtual workshop, which I think you may have heard talked about in some of the previous sessions, mitochondrial dysfunction, and NASA's HRP, the Human Research Program Investigator workshop coming up as well. All right, so now I wanted to start to cover these areas that we talked about, which are areas that the science community here has advanced a lot in the last 5 to 10 years that we feel could really help us in the space biology arena. And so there's 6 different topics here that I wanted to just cover briefly to give you an idea of some of these areas of research. So the first one is probably the most familiar to all of you with omics and systems biology. As we all know, the tools for omics and systems biology and quantitative genomics have really blossomed in the last 5 to 10 years, with increasingly more tools available for us to do this research. And so NASA, in response to that, we have, for example, developed the Gene Lab database, and you may have heard several talks at this session related to the use of that database. But it's where we archive space-relevant omics data, where folks in the community can go in and download or analyze the data. There are tools there that help with the analysis, and you can upload your own data there, for example, and compare with some of the spaceflight data. And there's also an analysis working group that works together. And there are several people there who've never worked with NASA before, but this data is openly available for everyone to use. And then related to open science there again is that second system there called the Ames Life Sciences Data Archive, or ALSDA. This is something we've had along with the LSDA, which is the larger NASA archive. NASA has had this and supported this for many, many years now. And part of this ALSDA system is the fact that we also offer tissue from spaceflight or spaceflight related studies from a variety of different model systems that's open for the community to request samples so that you can analyze those specimens for preliminary data, or after your spaceflight, if there's something that you want to compare against, you can request samples from that. And we're in the process of seeing how we can just update this particular archive, which is an old database. So then going to the systems biology aspect, and what do we mean, or how will it relate to helping us in space biology? So again, this idea of these multiple stressors that impact biology when we're in deep space, one way to start to understand what some of the underlying changes and what the commonalities are between different systems is we can use model organisms of increasing complexity, starting from yeast to worms to flies, mice, et cetera, and start to see what some of the common themes, you can use cells, organ systems, whole organisms, and then start to develop a systems biology model to see if we can start to predict some of the physiological changes that we anticipate in this environment. And to do some of this work, of course, we could utilize our ground-based systems, which include the radiation facilities that we have available, including places like Brookhaven National Labs, as well as some of our simulated microgravity and hypergravity facilities. And then couple that with the spaceflight studies, starting with some of our low Earth orbit capabilities, and then hopefully develop our experiments and mature them for longer term experimentation in deeper space environments. In terms of quantitative genomics in this arena, in this first topical area, again, on Earth, we have really been able to utilize this tool to understand the genetic basis of complex traits in different environments. And so using, again, well-characterized, well-established tools for quantitative genomics for different model systems that we have on Earth can really help us look at the variation of traits, genetic traits that are relevant to the space environment, to start to come, to use things like GWAS, et cetera, to start to come and utilize and predict the more robust systems that we can start to develop for use in the space environment. And we had a workshop on this topic that we held earlier in the summer. So the second of the 6 topics that I wanted to cover is genetic engineering in plants. So this, again, the tools, the omics tools that we talked about, are not just applicable to animal work, but of course translate very effectively into the plant arena as well. And so, in the plant arena, doing predictive breeding is something that we as scientists have been doing for a very, very long time. And using some of the tools online now with CRISPR and, all these different gene editing and other tools, and the omics tools and so on, we can really start to, again, understand what some of the basic underpinnings are for plant biology, so that, again, we can consider some of these following, and not only understand the mechanistic changes, but begin to look at things like, for example, how can we develop plants that are of compact size, or give us high yield, or have a high level of some particular nutrient that would be valuable for our astronauts to consume. Or, for example, to start to look at plants that are hardy and more robust in terms of withstanding very dry, desiccated conditions, or, elevated carbon dioxide levels, and so on. So the third of the 6 topical areas that I wanted to discuss is this idea, again, of utilizing regolith for living off the land, so to speak, and being independent when we go into the moon and the Mars environments. And here I wanted to point out that some seminal work was done, back in the Apollo years in the 1970s. And you heard a really nice talk, I think, at the opening at the President's Plenary earlier this week, that really described some of the changes and some of the science done on the surface of the moon. And some of the early studies actually showed that plants could be exposed to lunar regolith without any negative, any large discernible negative effects, and that plants could actually uptake nutrients and minerals from the regolith. However, it is also important to understand that these were very limited plant growth studies, because that lunar material was very limiting in quantity and very precious, and still is. And also they were in contact, the plants were in contact with very small amounts of this material. So really we have a lot more that we need to understand about the effects and how we can effectively utilize this material, presumably to help us with plant growth and to live off the land. In this context, I wanted to also bring up this idea of using microbes. We can utilize microbes to help us make some of that regolith more usable for the plants, as well as to use it for bioregenerative life support in terms of the ability to produce, oxygen, sink carbon dioxide, and recycle some of the organic and inorganic materials. So now the animal responses to lunar regolith. So again, back in the Apollo years, some early studies were done to show that protists, invertebrates, and fish could be exposed to crushed lunar regolith without any apparent negative effects. So that was great. However, several studies over the next decades, however limited they are, did start to provide additional evidence that perhaps there are some effects that we need to think about in terms of animal health. And really, as we now have more tools at our disposal to look at detailed physiological, cellular, and molecular changes, there begins to emerge some evidence that maybe there is more to the story than we understand at the moment. So, for example, rat inhalation studies were done to show that there was a concentration-dependent increase in some inflammatory biomarkers seen in the bronchial alveolar lavage. Some benchmarking studies were then also done with rats to start to try and estimate a PEL, or a permissible exposure limit for humans, in terms of how much dust they could inhale, over a period of time in this, in the lunar environment, for example. Again, those were extrapolations from dust and only looking at the effects in the lung. And then lunar dust can also is thought to be a mild irritant for the mammalian eyes. Now, as lunar material itself is limiting, some more studies were done with simulants, which gave us a little bit more of a picture of other changes that might occur from some of this material. And so, for example, the first study cited over there looks at rat synaptosomes. And what it finds is that when presented with these micro nanoparticles of dust, there is an increase in the binding of glutamate to the nerve terminals. So you can imagine that if this dust were to impact the central nervous system, messing with the glutamate homeostasis in the central nervous system could lead to functional consequences. And also, when you compare with the idea that we have in the literature now, quite a bit of evidence that radiation can affect the nervous system and can affect both structurally and functionally, can cause impact with ionizing radiation. So, again, this idea of combined stressors. So when you have multiple stressors that could impact some of the same organ systems, it becomes interesting to see what the ramifications of those might be when you're exposed to those multiple stressors in space. Similarly, some in vitro studies with macrophages were done to show that there's a concentration-dependent increase of inflammatory markers in mammalian macrophage cells in culture of iNOS. Lunar simulant treatment of neuronal and lung cell lines resulted in cytotoxicity and also DNA damage in both genomic and mitochondrial DNA. And then some rodent studies indicated that intratracheal instillation could cause some inflammatory damage that could then indirectly affect inflammatory myocardial fibrosis, as well as, with the infiltration of lymphocytes and neutrophils into the lung, could cause inflammatory pulmonary fibrosis. These are just some, just from literature searches, and there are possibly several other studies. But really, the message here is that we need to gather more data on both the plant growth, how we can utilize regolith material to our advantage for plant growth in these environments, as well as to look at whether there are any consequences of exposure to these materials to human health that, again, we can protect against, but we need to know, understand, so that we're ready. The fourth of the six topical areas that I wanted to cover today is this idea of 3D tissues and advanced microphysiological systems. And just to give you a sense, the first organ on chip, the lung on chip model, was reported by a group at Harvard in 2010. 11 years later today, in 2021, we now have 12 such organ systems that are readily available for purchase, for example. So some of this work has been progressing very rapidly, and in fact, thanks to a lot of funding from the NIH and its various institutions, including NCATS, as well as the FDA, DARPA, and EPA. And you will see on the next slide, actually the slide after, that NASA also has been, actually no, I talked about it earlier with our solicitation that's out there on 3D tissues. This is also an area where we're beginning to do some ground studies to look at the effects and utilize it for looking at the effects of multiple stressors. And so in terms of space biology application, again, the idea would be, the idea would be that being very complex systems, and we know that we still have a lot of things to work out with this relatively new technology, the idea is that we would get robust model systems developed on the ground, make sure we validate that they're relevant to spaceflight applications, and then utilize, for example, first in low Earth orbit on the International Space Station or on commercial space stations when they come online. And actually the ISS National Labs has also been doing a lot of work in this arena, working with the commercial sector and with the academic part of the science community to begin to realize some of these changes as we move forward. And then the idea would be, once we have these tested in low Earth orbit and we see and we validate them, we see the relevance, and then we can start testing, using these to test multiple spaceflight stressors using human cells and human tissues in these systems. Another added advantage could be that if once we get these systems going, we could actually use them for personalized models with these 3D tissues. So for example, we could take iPSC-derived cells from astronauts. We could create these 3D tissues and organ systems and then test them either on the ground or in spaceflight with these different stressors to start to see how would a specific individual respond to a certain environment, again with that idea of preparedness, so that we know and we anticipate what the changes might be and we can be ready for them on an individual basis. So to integrate these first four areas that we talked about and the research that I've talked about so far, the idea then would be that we do work with a variety of model systems, starting from single cell systems to multicellular simple systems to more complex systems, starting from the single cells to, worms and flies and mice and all of that, as well as with plants, model plants and more complex plants, and begin to integrate this information to to come up with some predictive models of what to anticipate. And on the other side, then we can take human cells and human tissues and utilize these 3D systems to start to see what the effects of these multiple stressors would be. And then together, then the hope is that with the data that we collect in our fundamental research program, we can start to input and create a story that will then help us, NASA, and the community get to these environments that we want to explore next. The fifth of the 6 areas that I wanted to cover, and sorry, this is an eye chart, but is artificial intelligence and machine learning. And I think one of the reasons it's an eye chart is that if you just look at the underlined section even over there, the market growth that we anticipate in this industry from this year being approximately $93 billion US, in about 7 to 8 years from now, we expect that to be close to $1 trillion. So, I'm sure I don't need to, reiterate to any of you how much we use AI/ML and how quickly these tools are developing. as we use it for image analysis, we use it for wearable health devices, we use it for natural language processing, and the idea is how can we use it within NASA? And so this past summer and the summer before, NASA's space biology program partnered with NASA's human research program, and together we funded this Frontier Development Lab, FDL. It was a challenge where researchers from academic institutions worked closely with companies like Intel that have been developing these AI/ML tools for their purposes, and then partnered with groups like the Mayo Clinic for disease database data to see how we can start, and then using the data from GeneLab to see how we can utilize all of this information to come up with some causal inferences and start to predict some of the changes that we anticipate. And again, you might have heard Sylvain's talk in the last couple of days that talked a little bit more about some of these tools that one can use. We held a workshop for this in the June timeframe this past year. And again, in terms of space biology application, we can use AI/ML tools for, looking at multi-omics data, as well as automating, imaging, analyzing image and interpreting imaging data from space flight. Remember that as we go further into space, it will get harder also to communicate and send large batches or large volumes of data. So sending raw data might get harder and harder as we go further away. And so having ways in which we can automate the analysis of this data in deep space, sending back at least, if not completely analyzed, but at least partially analyzed data could be an advantage. And not to mention things like, live diagnostics and monitoring for astronaut health when, for example, they're in the Mars environment. And various other applications for space biology. The last of these topical areas that I wanted to cover is this idea of automation, miniaturization, and data telemetry. As a whole, we've all noticed, how much smaller medical equipment is getting, laboratory sampling tools are getting, and the increase of the use of telemedicine tools. In our time of the pandemic, we don't need to look very far to see how much these tools have developed and will continue to develop. And for spaceflight, for our purposes, miniaturizing, automating, when you're doing an experiment on the lunar surface where you don't have much crew intervention or even on Gateway, and definitely not when we're, doing our experiments in Mars, using these tools and developing the capabilities could become very useful for us to do our science. And then telemetry, again, as we were saying, sending data back gets harder and harder as you go further away from the surface of the Earth. And so we held a workshop for telemetry-based biology in August of this year. How would we utilize it for space biology? Well, one of the ideas that we have and that we're looking into is looking at these science-focused habitats. So many of our commercial partners in low Earth orbit have been working with us to devise habitats and equipment and tools for use on the International Space Station and in low Earth orbit. So how can we begin to utilize some of these capabilities in deep space where, again, we have the lunar day and the night, and so, how do we maintain power during the dark period, for example? How do we protect the habitat from the onslaught of some of this radiation? All of this put together, coming up with a modular system, excuse me, to accommodate multiple types of organism, do autonomous readouts and data telemetry becomes important. Craig mentioned the importance of the decadal survey, and I just wanted to stress here again to this audience that the research campaign papers are due on December 23rd. And the decadal committee recommendation becomes a very important influence to focus space biology research. And in terms of research campaigns, these are these large efforts which could be, up to tens of millions of dollars to do some of these very ambitious projects that will help us in our effort to get into deep space. So please do put in those white papers. Partner with groups, whoever you think, to really get the message out there for the kind of science that you want to do and to advocate for it. In conclusion, I want to say that as NASA plans to return to the lunar surface, develop a sustainable habitation there, and then to utilize that to piggyback that, what we learned from that, to go into Mars, we in space biology intend to utilize multiple biological model systems and many different spaceflight platforms so that we can understand the mechanisms of change in biological systems over long duration exposures to deep space, so that we can really enable exploration. And of course, as we've always done, the research that we do then, and really all research, I think at the end of the day, gets to benefit life on Earth, both in terms of human health as well as in agriculture, for example, in controlled environment. And with that, I will stop and thank you very much. Thank you. And now I wanted to introduce Kevin Sato, who needs no introduction, I think, to this crowd. And Kevin is the program scientist for exploration, and he'll tell us more about the space biology connections with the exploration missions. Thanks. Yeah, so this one is forward, back.
Okay.
And then this one, the blue one, is
Laser
the
pointer. Great, thanks. Well, welcome everyone, and thanks Sharmila for having me speak. What I'm going to be talking to you today is just an overview of what BPS is currently doing in order to really understand and to bring our science into the realm of beyond LEO, as well as into the areas that are with groups that are actually designing and developing the vehicles, the capabilities, and the resources that we need to do our research. So as you know, for over 20 years, we have been conducting research in low Earth orbit in an area that can only be conducted and not possible on Earth. But as we begin to start our adventure out beyond LEO, we'll now begin to study research that cannot be conducted in low Earth orbit.
3, 2, 1, 0. Ignition. Liftoff.
Artemis 1 will lift off from Launch Pad 39B at NASA's Kennedy Space Center in Florida with 8.8 million pounds of thrust provided by the most powerful rocket in the world, our Space Launch System rocket, or SLS. The uncrewed flight will be the first integrated test of SLS, our new Orion spacecraft, and exploration ground systems at Kennedy. Artemis 1 will send Orion beyond the moon, 280,000 miles from Earth, farther than any human spacecraft has ever flown.
This is not NASA doing this. This is the United States of America doing this program, the Artemis program, and there are companies all across our country that have a part in it, so there is kind of this wave of excitement being generated just by saying, we're going back to the moon.
After the upper stage of the rocket separates from Orion, the upper stage will deploy small satellites over several days to perform science experiments and technology demonstrations. Orion will make its multi-day outbound trip to the moon, propelled by a service module provided by the European Space Agency. Engineers will test Orion's systems on the way to the moon. Then, Orion will fly about 60 miles above the lunar surface, using the moon's gravity and engines on the service module to enter a lunar orbit. After about a month and a total distance of over 1 million miles, Orion will return home, faster and hotter than any spacecraft has before. A primary goal of Artemis 1, ensure Orion safely returns to Earth before we fly with humans. When we do, we'll build our capability for sustainable lunar exploration, preparing us for missions farther into the solar system.
Initially, what we'd like to do is start establishing a presence on the moon. So we're going to establish going back there on a regular basis, and then we'll end up setting up Gateway, and we would launch to the Gateway, and from Gateway, land on the surface of the moon.
We are there for, weeks, months on end. And there, we're going to be able to test out all the hardware and the habitats and the hatches and the suits and the rovers that'll allow us to prove out those technologies. The moon will lead the way to Mars, and we should be there within the next couple of decades.
Right. How do I go back? Got it. So with that video, that begins our adventure, which will hopefully be in February of 2022, which is currently the launch date for Artemis 1, in which we'll have our first science experiment on, as Sharmila has discussed in the past. What I want to show you is this is a snapshot of what Artemis is. Artemis is just not SLS and the Orion capsule. It is a constellation or suite of platforms that will take us to the moon to enable us to do scientific research, including SLS, Commercial Lunar Payload Services, the Gateway, Orion, the human landing systems, as well as eventually Artemis Base Camp. This is the standard swoosh map that I think you've all have seen already, but it really represents for now the general plan for how NASA is going to return to the moon, starting with Artemis 1 and then leading to the first landing, where the first two humans will take their first steps back onto the moon. In fact, this will include the first woman to step on the moon, as well as the beginning of the development of the orbital gateway. As we begin to progress past that, we will then begin to develop Artemis Base Camp, in which the gateway will be fully built. It'll have two habitat areas called HALO and IHAB from ESA. It will also utilize a logistics module, similar to what we used for the International Space Station that will carry the bulk of our experiments as well as other supplies. And also it will serve as the gateway by which the human landing system will return to the moon continuously. Again, as you see on the lower areas with the unmanned CLPS vehicles, those will be continuous. There will also be a rover that will extend traverses, as well as eventually a pressurized rover that will allow for much further and extended missions and durations of times outside the landing systems and the habitat, and finally the sustainable habitat in the end, with the goal of 4 crew members being down on the moon. In terms of the lunar orbit, this is the gateway. I think we've heard a lot about the gateway. There have been workshops that have been conducted on the gateway, and it will be our ability to get to the moon, but also it will have laboratory space on it for us to conduct research, both within IHAB, which is the European, as mentioned, habitat, as well as inside of the HALO. Also, there are going to be internal and external SORI positions, or positions in which experimental platforms can be placed. The first two are Ursa and Hermes. Ursa is a radiation monitor instrument that will collect incoming radiation from deep space in the sun. Hermes is a heliophysics space weather instrument to understand the physics and the chemistry of the sun, but also it will be collecting data. All of that information and data will be available to our science community. IDA is actually the internal dosimetry array. It will include arrays from both ESA as well as NASA. So we'll have the opportunity to see both the incoming radiation from outside of Gateway, but also the radiation that is actually incoming after it passes through the body of Gateway. Gateway is an international team. It's currently NASA, CSA, or the Canadian Space Agency, the European Space Agency, and the Japanese Space Agency, who are currently working to develop this and to conduct the first series of research that will be done. This isn't a slide I made. This is a slide that was made by the Artemis team, and it is one that recognizes the research that we need to do. This is unique. In the past, I think you've all been aware of the fact that we've been an afterthought. Science has been an afterthought to any vehicle that's being built. But today, it is actually part of the main process by which they are going to be developing these systems. And there are utilization groups that BPS is embedded in order to be able to provide our research needs and capabilities in order to make sure that our science can be conducted. So we're not at the end phase, we are at the beginning phase. And so again, life sciences, physical sciences, fundamental physics, and in this case, food and drug degradation is one they added. But you can see that at a very high level, our science is already in the minds of those who are doing it, and we do have champions in areas that are in the areas of technology and vehicle development for conducting our research. In terms of what we're looking at, or what space biology is looking at to a certain extent, is really the hazards of human spaceflight. And these were 5 hazards that were identified by the Human Research Program and the Human Systems Risk Board as the key areas of which we need to conduct research, both fundamental and applied and translational, in order to ensure the safe travel of humans in spaceflight. And that's just not necessarily for our own astronauts. That's also for possibly all of you who may be going to space, as well as for commercial and other aspects. We need to make sure everyone is safe, and by that, we need to do that by conducting our research. As Sharmila has mentioned, we are not going to just be isolated to a single platform. We are working to make sure that our science is on every opportunity and every platform that's going to the moon, including the Orion capsule, the human landing system, the Gateway, as well as commercial lunar payload services. But keep in mind that we are not going to leave low Earth orbit. It is still an area of extremely critical research that we need to do. And so the low Earth orbit platforms are still going to be used, such as ISS, Bion, Free Flyers, the new hopeful space stations that will be out there, in order to continue to conduct the research that can only be conducted in LEO, but also now to possibly serve as controls for research that's going to be done in deep space to understand the unique elements of deep space and Earth. So as you're doing your research, as you're looking at your designs for science, don't forget the International Space Station and low Earth orbit, as well as ground studies, are still extremely critical to conduct research in deep space, as well as proving grounds before you actually design and take your science out into space, but also for any new technology that you will be developing on your own. So we look at it as both location-specific science, but also a continuum of research across all platforms and all locations. And that was critical to TIDES and what Sharmila was describing earlier. Now, in terms of what has been and what is going on, as I've mentioned, we're embedded in all of the areas of scientific utilization that is being used to develop the high-level Human Exploration and Operation Mission Directorate documents, or the HEO documents and the AES documents, that are being used to drive the requirements for facility use. So questions that you had related to, are there going to be human habitats, and is a laboratory going to be developed and designed for our use on the moon? Is the unique needs of our community going to be included in Gateway as well as everyone else? The answer is yes. There is language that is being put in stating that there is a physical sciences lab that will be needed. There is a life science laboratory on the moon that's going to be required, which includes glove boxes, freezers. It also includes everything from microbiology, plants, all the way through to eventual rodent or vertebrate research elements. So, and the interesting thing is we're not hearing, no, you can't do that, or you can forget about that. What we are hearing is, what do you need? And then we're hearing that information from those who are developing in the leadership for these areas, perpetuate that information up through the chain. And so our science, again, is being heard. But what I would recommend that you all take a look at is actually read through the Artemis 3 science. Although this is the discussion for the science that is being recommended by a science definition team for Artemis 3, it also contains the high-level objectives that are actually being used to try to define what we need to do for research into the future. And that research has been defined from past studies, as you've seen from previous talks, but also reviews of the current objectives that were out there by our program scientists, both in fundamental physics, physical science, as well as in space biology, to make sure they are current at a high level. Right, and this is just a short description of Objective 7 within the investigation and mitigation exploration risks that we have. And as you'll see at a high level, our science, this community science is there, and it's all being included and added in. This is not considered a stale and dead set of requirements. The objectives that you are all, or the white papers, excuse me, that you are all putting in as topics and campaigns for the decadal white paper are going to be used to actually update these objectives as the program Science for Space Biology begins to review those priorities and recommendations in order to understand, is this still current? What do we need to change? What do we need to do to make things that are new? So that's why this is extremely important that you submit and that we've received white papers, because they are going to drive what we do, but beyond that, it's going to drive the capabilities that we're going to be provided. In terms of funding opportunities, there's a lot possibly out there. And what I want to stress and emphasize to all of you is that you please, if you haven't already, and if you're only subscribing to notifications from BPS, that you do a general notification from as many of the different SMD divisions, especially Planetary Sciences Division, because within the Planetary Science Division is where notifications will come out for CLIPS payloads. It's the area that you'll see potentially areas for lunar science that may be of importance or interest that you can contribute to as well for their calls. So again, don't just focus on the BPS science notifications, widen your reach beyond, because also there are Solar System Exploration Virtual Research Institute cooperative agreements that are also coming out that will likely have experiments that you may be able to contribute to as being part of the research. The one thing I did want to mention is, as we've mentioned, are the PRISM calls. And these are the calls for CLIPS. And they come through the Exploration Strategy Science and Integration Office. There's one that's currently out, PRISM 2. PRISM 3 is likely to be released sometime mid-2022. They are one call each year for 2 future lunar landing missions on the CLIPS vehicles, therefore instrument development, as well as associated research for computer and science providers. Just to let you know that I have had a presentation and I spoke with all of the CLIPS providers to provide them the unique characteristics of our science and our needs, such as late load, sample changeout if there aren't flights, cold stope, power, a lot of different things that we need to do to keep our science alive or to activate it, something that they didn't have before, but they now have. And so they are incorporating that information into our work. So again, keep an eye open for a lot of the solicitations that are going to come out. And the other thing I will also want to stress to this community is this is a new area that you're going into, something that maybe some of you have experience in understanding it and others you do not. As you do studies such as in regolith, as you do studies on the moon, you're going to want to know what's in the regolith. Do you know what is in there or not? The lunar side of PSD is really saying we need to work with you. And I've talked with them, they've talked with us in order to integrate and work together on the science. So 1, you could do your research, and 2, interpret your data based on their aid in understanding what environment you're in, what is in that regolith. So also I stress that you all please look at Lunar Science Institute, subscribe to their notifications for their lunar surface science workshops, as well as the Lunar Exploration Advisory Group annual meeting. That is the lunar sciences community's complement to the ASGSR, but it's also going to be what I hope as part of our community's complement as well. So please attend and please learn more about the moon. We hope to actually start to integrate our topics and areas into the league as well. And they've actually reached out to Craig as well. Just to give you an idea of the CLIPS deliveries, the triangles represent the PRISM 1 calls location to Reinhardt Gamma, which has interesting electromagnetic fields, and then to Schrodinger Basin, which is PRISM 1b in the South Pole. The green circles represent the 2 site locations in general for where PRISM 2, so the current call that's out there, where they're going to go, which is up to the Garthusian domes, as well as a location in the South Pole, which still needs to be defined. All right. And so as we're doing a lot of the work on the moon is a place where we're going to go to stay, where we're going to sustain research, and we're going to hopefully do a lot of things in partnership with many people. But the next giant leap that the moon also represents is going to Mars. And this is a picture of on Mars by Curiosity looking back at Earth. Activities have started and have been ongoing for research and utilization of science and vehicles for transit to Mars and on Mars. And so that work is currently going on, and BPS is becoming embedded in that work and being communicated out. So just to let you know, we're just not staying on the moon. We are actively involved now in how do we make sure that our information is there in order for us to make sure that when we go to Mars, that we're not behind the 8 ball, but now they're making sure we're embedded now. And just to give you an idea of one thing that's been going on, Julie Robinson and others has been leading a group of internal NASA experts and scientists, as well as administrators with the ISS to utilize ISS for Mars. In other words, to see how we can use ISS in order to conduct early studies there to understand what transit's going to be like for Mars. What it's going to be like to have technically 40-minute comm changes, 20 minutes to and from return, and how can we make things more independent? Because as they're going to Mars, we're not going to be chasing them. There's not going to be someone there on Mars waiting for them to step off on Mars after they've gone on the transit, say, hey, we're okay. Or no one is going to be there to chase them down if there's a problem. There's going to have to be a lot of automation. And so one of the areas that they're looking at is to actually use ISS as a Mars transit analog to conduct research. Now that's not going to, the key though is it's the clear understanding that there are also critical research and other things that are going on ISS, so it cannot interfere. And that was one of the major tenets for this, is cannot interfere with research that is currently going on ISS. Okay, and so that's what we're currently working on. And so what I like to do is just the final video.
Ignition sequence start. All engines are running.
We have taken tremendous steps.
We choose to go to the moon before this decade is out.
We have achieved the earth-shaking, the breathtaking, the groundbreaking,
one small step for man,
and left a mark in the heavens. Our successes build one upon another and amplify what is possible.
The dawn of Orion.
It's time we take the next great leap. We're building the next chapter of American exploration, returning to the moon to stay, so we can go beyond to Mars, to expand what's possible and further our understanding. The architect
is going to go long, and we want to make sure we have time for questions and answers, so we can show this afterwards as we get set up for the next set of talks. So Craig, we'd like to go ahead and start our Q&A, and we'll go ahead and show the video in a sec. Thank you.
All right. Mantha, feel free to come on. All right, so, all right, I'm going to start with the questions now that you guys are all seated. So the first question is, has NASA defined experimental regolith? I think by that, the question is, do we work with simulants? So let me try and answer that firstly, if you have questions, please text it to 1-855-855-1413. So in terms of regolith, so there's simulant that's available commercially, so from companies. JSC used to make simulant that was provided to the science community, and I think they've moved some of that technology to for some companies who now can provide. So simulant is one thing for both Mars and lunar simulant. In terms of actual regolith sample, one of the things you heard from Kevin's talk that we are planning to go back to the surface of the moon as well as surface of other, Banu and all of this. So we are hoping, even though those samples will also not be in very large quantities and will of course be somewhat limiting, but there again, our hope is, and we will see how it evolves as time goes on and as some of these missions begin to bring back regolith, that once we do these initial studies with simulant, that we start to see what some of the compelling things are that we need to do where we request a regolith sample. All right, so a question for Kevin. Can an update be provided on the progress of Gateway and whether it's at risk of being canceled?
Sorry. It's on. Yeah, so the current process for Gateway is that they are preparing PPE and the Halo element to be launched together, as well as IHAB is continuing to be developed for launch as it relates for the European Space Agency. So it's continuing to move forward. Dates in terms of when Gateway will be initiated with its first launch of the PPE and the Halo are still in discussion, so I don't have that information. However, Gateway is not going to be canceled. It is continuing forward as a critical element in our return to the moon and sustained utilization.
Thank you. All right, another question. This one is for me. It says, traditionally, space biology focuses on fundamental biological science related to spaceflight. Your wonderful talk focused on translational science, which is valuable for the future. Trish also concentrates on translational science, and so what's the main difference for the focus for space biology and Trish? Good question, and so there it's important to remember again that we are a fundamental research organization or program, so we have a lot of flexibility in the organisms that we can use. Trish typically focuses, because it is more closely related to the human research program, focuses on systems that are very directly applicable to human risk assessment. We, on the other hand, are free to utilize any model systems that we can feasibly and practically get into space that answers a question that you identify, or we as a community identify, as being relevant and important for long-term sustainability in space. And so we have the greater breadth of the science that we can do. We have a greater breadth in the tools we can use. We don't have to be applied necessarily in terms of what we create. We don't have to necessarily come up with a tool or technology or something that's applied in the immediate term. We really can focus on the larger fundamental research. I think actually, let's see. All right, another question. How do BPS or SMD think about combining ground testing versus suborbital microgravity testing to gain flight heritage for their experiments ahead of the mission? Again, I can take a stab at this, or unless anybody else would like to. I'll go for it. Okay, so I don't see it as an either or one versus the other. I see them as all tools that are available, and it really depends on the science question you have. So for example, a ground study might be very useful for radiation, for preliminary radiation studies, doing GCR, mixed beam simulation at Brookhaven could be very valuable to set up your system. On the other hand, you might have and which, for example, for that, the suborbital platform may not be as handy just because it's, like a few minutes of microgravity and it's a short flight, right? Whereas you might need longer or fractionated doses of radiation to really start to see the effects that you want to delineate. On the other hand, the suborbital becomes very valuable for example, technology. You know, if you're testing an equipment or an experiment that you want to make sure would work in microgravity, so you can do a hardware validation or an experiment validation. Or if you're looking at a change, you know, whether it's transcriptional or some kind of a signaling change with a reporter molecule, which you know happens in a very quick time frame. It's a transient or a transitional change that you know you will pick up in a short period of time, then the suborbital becomes a good platform to validate. So I see it more as these are all the tools that the aerospace sector, coupled with NASA, coupled with the international community, coupled with, you know, ISS National Labs, et cetera, are providing. And so it really is up to the science that you want to do. All right, other questions? No speaker mentioned planetary protection in the Moon to Mars strategy. Was this an intentional deletion of an international obligation? Not at all. In fact, you heard some planetary protection talks earlier at this conference. And so absolutely, I mean, any mission that we do, now as you know, on the Moon, planetary protection is not as worried because that environment is not thought to be conducive to growing some of the Earth-based organisms. So there's definitely not you know, as much concern for lunar experiments, but of course there is going to be eventually for Mars exploration, and planetary protection is just something that every mission does. So it's not, definitely by no means any intentional you know, leaving out of the discussion. It's just something we take for granted. We have to do it. We do it. We work actually closely with the Planetary Protection Group, and this is, you know, and you heard them present at this conference. So I hope that answers that. This one, if you want to take this, Kevin, as NASA and space biology develop new payloads, where can PIs find the details on hardware to best design experiments for the upcoming deep space related solicitations?
Yeah, so for the clips, they're basically the information that's related to what you need to design to is within the calls themselves. I think as you've heard, unlike in the past where we were given ICDs, interface configuration documents, everything you had to fit into or a box, right? And that said, then told you, okay, fit your science in there, fit your instrument in there. For the clips, it's completely different. You develop the science that you need, and then you determine exactly what you need to conduct the experiment, either the instrument or the hardware. But what you don't want to do is you want to do anything that's super exotic, you know, that no one has. You want to stay common. So in that case, you're open to design and develop what you want. They will then, as part of the suite, if it's selected, will then send out a task to the CLIPS providers, and they will say, yes, we can build that in. They will build their area within their lander around what you need to interface, to interact, and to provide power and communication. As it relates to any of the other deep space elements, there's currently no hardware out there. It's really the hardware that we may already have in place or that we have. Now, there will be interface configuration documents as it relates to how Gateway will be used, or potentially HLS and others, but they're currently not available in general to the public. So the idea is if you are going to fly, you'll just need to wait for calls that come out through us or BPS, and then we will have information as it relates to the hardware that might be available, or we may have RFIs that are called for hardware development, which will then have that information in it.
Yeah, exactly. And just to follow on what Kevin says, so for example, for our near-term calls from space biology for some of the deep space missions, just because, as Kevin mentioned, we're limited in what we can and power allocation and so on. So at the initial phases, we will define in the solicitation what we can accommodate. So that will help shape some of the science that we're able to do. And it's kind of a progression. As we get more and more capability, we'll be able to utilize more and more sophisticated hardware. At the beginning, it'll probably be simple, but we will defined that in our solicitations.
Yeah, one other thing too, it's also the hope that any instrument that's being developed by, say, your lab, in the past you've had, hey, I got a piece of hardware I want to fly, and it's like, yeah, this time it's different. You are likely to be able to, but it will hopefully remain in the suite of hardware that other investigators can then select and use as well. So keep that in mind as too.
So this question, and I think maybe this might have to be the last one so that Mamta can introduce our next set of speakers. And this maybe, Craig, if you want to take a stab at this, the question is, what are the current plans for the ISS end of life?
So we're waiting for congressional legislation on this front, but all indications are that ISS will be authorized to go to 2030, and the international partners are in agreement with that. You may have heard earlier in the conference that as NASA stimulates the development of commercial LEO destinations, meaning commercial stations, that the aim is to start a transition starting in 28, so that there would be an overlap of 2 years where both ISS would be used and these commercial LEO destinations. And I think you can anticipate that prior to 2028, there might be some, pilot activities, which, BPS and our pioneering, mission would be happy to be part of. So basically, it means the latter half of the decade. We expect beginning of a shift towards operations with commercial LEO destinations, with that really picking up in 28 and culminating by 2030. And as Sharmila was saying, we are fully expecting to use LEO for a long time. I mean, it's the when you need long-term microgravity, that's the closest place to go to get it. And we don't need to go to the lunar surface if we're just focusing, like lunar orbit, if we're only focusing on the microgravity aspects of something. If we need the deep space radiation, then we have to go further out. But bottom line is, for a lot of the work we anticipate doing, LEO will suffice, and we'll be an active user of LEO for the foreseeable future.
Great, thank you. So I think in the interest of time, and really I think we covered a lot of the questions that are here, I will have Mamta introduce the next set of speakers.
Thank you, Sharmila, and sorry about that. So I think the next group of speakers represent something grander, and I'd like to give you just a little context before we bring up our first one. And that is, if you remember, in 2020, when the world changed, you may not have realized that about 3 months into that pandemic, we launched Mars 2020. And it was our first major science mission to persevere through what was happening before we knew how to handle it and get up into space safely. And so it would be no surprise now, as we look back, that the Mars rover is called Perseverance. Why am I telling you about Perseverance? Because I think the next 3 speakers, the future scientific leaders, are not just that, but they are representative of how we as a community, and I hope you do not lose this context, we as a community have persevered through 2 of the most tumultuous years. I know I had a low point in my own life, managing 3 young children over the last couple of years, while hunkering down and ripping them from their normal. They represent that perseverance. So with that, Dr. Aubrey O'Rourke is going to start our next early career session and tell us about microbiomes on the ISS, and her talk is titled Microbes from the International Space Station Built Environment.
Wow, thank you very much for that introduction. I am Dr. Aubrey O'Rourke. I am at Kennedy Space Center as a project scientist, and I was formerly a NASA postdoctoral fellow at the J. Craig Venter Institute in La Jolla. I'll be talking about microbes from the International Space Station built environment. This project was made possible by a Space Act agreement between the Alfred P. Sloan Foundation and NASA. MOBI premise and objectives are that we spend a lot of time indoors and we want to know the microbial community that's associated. Of particular interest is the prevalence of species that might affect human health. From terrestrial research into MOBI research, we know that in the indoor environment, these surfaces are colonized by dust, and this can be transferred by the inhabitants to surfaces in air supply. The built environment, using the space station as a case study, we know that the ISS has constant human occupancy, controlled environmental conditions, and routine microbial monitoring. Viable members of the space station have acclimated to these pressures, and microbes themselves have interesting ways of transferring their genetics through mobile genetic elements and mediated horizontal gene transfer, gene mutation, and DNA arrangement or loss. So microbial surveillance on the space station is of the air, surfaces, and water, uses standard culturing techniques, has been conducted over 12 years and 22 missions, and resulted in a large collection of bacteria. Primarily, the surfaces have Staphylococcus species at 70% and 80% recovered from the air samples. Burkholderia and Ralstonia species, on the other hand, are found in the potable water system at about 60% of the representatives collected from culturing. I'll be talking about the publications conducted by myself and another postdoctoral fellow, Mike Lee. Okay, so our workflow is pretty basic in terms of genetic analyses these days, but we culture the samples that we get from JSC, we do sequencing, we look at the genotypes themselves, we have some phenotypic assessments in parallel, and then we correlate the two together. So in summary, we can really look at neat avenues when we do whole genome sequencing. So our scaffolds that we did from de novo assembly of our genomes from these surface isolates, we're able to then take an astronaut microbiome sample and look at how these metagenomic reads recruited to the reference sample of our space station isolate of whole genome sequencing. So this is an overview of that process. And we saw from one astronaut's nasal microbiome sample that over time we could see before flight, maybe not so much representation in that astronaut microbiome sample for a particular isolate from the surface, but in flight we see, particular reads mapping to one of our isolates, a Staph epidermidis, which is commonly found on the skin, and then also for a Staph aureus that can be found in the back of the nose. So we're interested at this interplay that's going on where we have Staph epi up, Staph aureus up, and Staph epi down. We looked at this interaction, and so Staphylococcus species, particularly Staph epidermidis, can create antimicrobial peptides that could alleviate problems from Staph aureus infection in atopic dermatitis. We also conversely know that Staph epidermidis can exhibit its own virulence if given the opportunity. So we look at this in the context of our isolate that popped up in the astronaut nasal microbiome. And we see for Staph epidermidis that we're able to inhibit the growth of Staph aureus as well as a quorum sensing aspect of our Staph aureus. We also look to see if Staph epidermidis could cause cell lysis to keratinocytes or skin cells. And we found that for our particular sample in the nasal astronaut nasal microbiome, that it did lyse keratinocytes, but not more than that of a terrestrial reference strain. So in summary, we see astronaut microbiome metagenomic reads mapping to our space station isolate. So that shows that our astronauts are indeed microbial vectors. This isolate has the ability to inhibit Staph aureus growth. And Staph aureus commonly can be associated with MRSA. This is a particularly methicillin resistant, not a methicillin resistant strain in this case. So I'll shift gears to Burkholderia of the potable water system. Burkholderia sapacea and Burkholderia contaminans are found in this potable water system. They can survive, this genera can survive long periods of time in distilled water. They're commonly found in soil, and they can survive as biofilms in infected individuals. So it's very difficult for people with immunocompromised situations to clear these infections. They also have a number of mobile genetic elements through plasmids that often contain virulence factors. So we looked further into this. We compared our NCBI available, so database available B. sapacea genomes to our ISS-derived de novo assembled genomes, and we found that they're genetically distinct and most closely related to a food pathogen strain. So that's quite interesting because this water is what is used to irrigate plants as well. We looked further into what set these isolates apart, and indeed they were flexible genomic islands often found on plasmids. We further looked into these assemblies, comparing them and aligning them, and then finding what is indeed on the plasmids, and it pointed towards virulence factors. So we wanted to assess how virulent these strains can be. So we infected mouse macrophage at a multiplicity of infection of 7, so that's 7 bacteria to macrophage. This is a way that Burkholderia achieves dormancy in an infected individual because they can hang out inside the macrophage and then be dormant and cause an infection later on. So in terms of macrophage lysis by Burkholderia space station isolates, we saw no more than our terrestrial reference strain, meaning that they're still able to lyse the macrophage, but no more. And similarly, in terms of invasion and replication, the Burkholderia are able to invade and replicate within the macrophage, but no more than our terrestrial reference strain. Luckily, all isolates remain susceptible to all antibiotics used in clinical trials or clinical treatment. We did find that, as in other studies, biofilm formation is increased from these space station isolates and in comparison to the terrestrial reference. We also did find for the Burkholderia contaminans isolates, they're known for antifungal properties. We thought maybe it was a new compound. It is not. It's acidic fungin, but in this case, we're showing that it inhibits the proliferation of Aspergillus fumigatus, as well as has hemolytic properties, so the ability to cleave blood cells. In summary, we have conjugative plasmids harboring virulent elements. They're able to invade and replicate within macrophage. They remain susceptible to all antibiotics, and they readily form biofilms for the space station isolates of Burkholderia. In conclusion, astronauts are indeed microbial infectors. Whole genome sequencing is a very valuable tool to understand the functional understanding of these space station bacteria. And the ISS isolates of Staph epidermidis and Burkholderia display virulence, but less than or no more than that of our terrestrial reference strains. Future directions, and I think this is interesting for this community because I wanted to know how we can better do sequencing in space. So currently, myself and a team are funded to automate the sequencing process for delivery maybe as a payload in a CubeSat or on a PRISM lander. Also within Kennedy, I'm given the ability and opportunities to continue this work using simulated microgravity on the bacteria commonly associated from the water system of the space station. And also we'd like to look into and are funded to look at the effect of biocides in water systems and their effect on crop microbiomes. I'd like to acknowledge the team I'm newly a part of and also the prior team at JCVI, as well as collaborators at UCSD, and also Dr. Mike Lee, who is part of AIMS, I think some of you may know him, as the Blue Marble Space Science Research Institute. So thank you for your time. I'll take any questions.
Let's see, I think this question is for you. Okay. So Aubrey, were you surprised the bacteria were similar on Earth? How many other organisms tend to do worse in spaceflight?
Similar, oh, in terms of their phenotypic response, not terribly surprised because nobody's actively, that I know of, getting sick from drinking the water. Perhaps there might be rashes that arise, but they don't seem to be a huge problem in the astronaut population. I don't really know the details of that sort of data because they're not released to us. Genetically, they are different, but they're not any different species. They're still just different variants of that species.
Great, and one last question. Have you or anyone placed a labeled bacterium on station and watched its spread or distribution over time?
That's interesting. I think we'd want to do that in a very controlled way. I think that is an interesting avenue for synthetic biology. So you could put sort of a marker in so that you know that that is indeed your bacteria, or even a kill switch so that you make sure that maybe there's an auxotrophy or something so you can control its further spread and it just doesn't become dormant or a resident of the space station.
Wonderful. Thank you so much. For our next speaker, I'd like to invite Dr. Natasha, and Natasha, please forgive me if I say your last name wrong. Haveman? Okay. Dr. Natasha Haveman, who's going to talk to us about using the MinION for plant growth studies in space.
Thank you. Good morning, everyone. So yeah, I'm really excited to be here. I want to thank the organizers for giving me the opportunity to do so. I thought I might just introduce myself a little bit, since I'm kind of new to the community. I am born and raised in Singapore. I came here to do my master's in translational medicine at Harvard Medical School. And when I graduated, even though I had an awesome experience, I felt that something was missing. So I decided to switch gears and join the UF Space Plants Lab with Dr. Annalisa Paul and Dr. Rob Farrell. And my experience as a whole in that lab has kind of solidified my desire to want to make this my community to establish my career. I had an amazing experience. I was able to do outreach work, participate in parabolic flight campaigns, which was an amazing experience. It's kind of up there with having kids. And I got to go down to Kennedy Space Center, watch launches, suborbital launches as well. And all in all, it was a really wholesome experience. And so for all the students in the room, I would just like to say, find yourself a great mentor because they can have a profound impact on your life. And in my time, I was able to participate in a lot of different spaceflight experiments. I did transcriptomics, suborbital transcriptomics, epigenetics, and the one that's kind of stuck with me or really dear to me is the Tejas spaceflight experiment. I spent 5 years of my research career trying to discover the function of an unknown gene from that data set. And I thought I appropriately named it OMG1. So now when I graduated, I was awarded the Space Biology Postdoctoral Fellowship, and I teamed up with Dr. Jamie Foster and Dr. Van Kat, and basically our research was to try and develop a method to look at plant microbiome interaction. Basically, for that particular research, what I did was develop a pipeline to isolate microbial RNA, get those RNA and sequence it in the MinION sequencer platform, which has long-read sequencing capabilities, and then discover what the microbial community was like, who was there and what they are doing. And so from our research, we were able to really, consistently between 3 different ladder samples, identify a very consistent microbiome, so basically who is there in the community. And then we were also able to identify different pathways, what active pathways were in the bacterial community and in the fungal community. So I was also involved in another project. I teamed up with Dr. Sugar from SLSL. And in that time, what we were doing was to try and diagnose the fungal pathogen Zinnia, the Fusarium oxysporum fungal pathogen. And we wanted to use the MinION sequencing platform as a tool to do that. So essentially, what our preliminary data was to excise the plant tissues that had the fungal pathogen, extract genomic DNA from it, and then use the MinION as a diagnostic tool to identify that particular pathogen. And we were really successful with that. I think preliminary data shows great potential. So why is that important? Because as we establish a sustainable habitat away from Earth, we will be taking plants with us. And it's not a matter of if the plants get infected with the pathogen, it's when they get infected with the pathogen and what we're going to do about it and how we're going to identify that if we can't come back to Earth to do sequencing and everything like that. And so that's not something new. The ISS was infected, like I said before, with the fungal pathogen, Fusarium oxysporum. And so we know that this is going to happen, and we need to find a strategy to solve these issues and mitigate any problems in the future. So this is where I would like to take my research as an early career scientist. Just this year, I was awarded a grant to do just that, basically looking at different plants, different phytopathogens, different symptomatic tissues. How would we extract tissues from these samples so that we can accurately diagnose them with the MinION sequencing platform? And the other aspect of it is to automate this system so that we don't have to spend precious crew time doing such laborious techniques on station. So I've teamed up with a few people, a few companies, and also with JPL to use their automated nucleic acid extraction processes, hopefully get the Voltrex to do the library prep and then use the MinION as a sequencing platform. So with that, I would like to thank all my mentors, my collaborators. One of the best things about doing this postdoctoral research position is you get to collaborate with many people in the room and to get a good feel of what it is like to work at NASA and all the background things of how research gets done. So I'd like to thank everybody and my funding sources. That's it. I'll take any questions.
Thank you, Natasha. First question for you. Could the MinION be used for any type of sample, or is it more specific to plant samples?
Oh yeah, it can be used for a lot of samples. Right now, I think they have been done with microbial samples. I think they're going to plan to use it for microbial tracking 3 to look at astronauts' microbial systems and the microbiome and stuff like that. So yeah, you can use it for any kind of samples. All you need is to get the genomic DNA out, or even RNA.
And then we have a couple minutes. If anyone else does have questions, you can text it to the number on the screen. And meanwhile, I'll ask you a question. You mentioned early on, you gave us the personal anecdote that you made a switch after coming here thinking that you wanted to study one field and switching to another. Do you have any advice for students who may not know what they want to do and how to sort of figure out what it is that they want to do? What kind of advice would you give to them?
Yeah, that's a good question. I mean, it's different for everybody. Personally, I think it's worthwhile going into something you think you're interested in, exploring it, see, how you like it, and then if you really like it, stick with it. If you think there's something missing, like in my case, I felt like it's not, hitting everything that I really am interested in, I'm passionate about. So then I decided to try something else. And so don't be afraid to try something else, I guess.
I think that's beautiful, by the way. Thank you. So last question for you. What omics analysis do you do?
What omics analysis do I do? So specifically for the MinION, I've done metatranscriptomics and metagenomics, but in my doctoral studies, actually, I've looked at transcriptomics, I've looked at epigenetics, epigenomics, and yeah, I think that's basically about it.
Wonderful. Thank you so much, Dr. Haveman.
Thank you.
And our last talk to round out this morning session is by Dr. Anna Simpson, and she'll be telling us about whole genome sequencing of ISS environmental microorganisms that enables the description of novel microbial lineage and prediction of AMR functions.
All right.
Well, hello, everyone. Thank you for sticking around. My name is Anna Simpson. I just started a postdoc this year at JPL in the Planetary Protection Group in Dr. Venkat's lab. So thank you for giving me this opportunity. And Aubrey has already talked a lot about whole genome sequencing and why it's awesome. So that means I can skip a lot of background. But today I'm going to tell you about the Microbial Tracking 3 project, which we're working on with our collaborators at UCSD, Jack Gilbert and Megan Timmis. This button? Oh, the green one. This one? Oh. So I'll start with an overview of the microbial tracking missions. So these, we've had microbial tracking 1, 2, and 3. So microbial tracking 1 took place from 2015 to 2016, and microbial tracking 2 took place from 2017 to 2018. And these are studies of the microbiome of the ISS. And so astronauts collect samples from 8 different surfaces. And so the missions have involved a lot of other sample collections, such as sampling of the astronaut microbiome, but those 8 surfaces have been consistently monitored over those years, and so we can compare back to that data in Microbial Tracking 3. So our basic sample processing is the astronauts wipe a surface, we receive that wipe back on Earth, we shake it up in buffer, concentrate the buffer, and then use that concentrate to do DNA extraction for qPCR and metagenomic sequencing, but we also use it to isolate bacteria and fungi from those surfaces, so we can do whole genome sequencing. And so we use 3 different types of media to target bacteria, fungi, and human commensals. And I want to note that this selection process isn't necessarily random. We can only select up to 5 isolates per media type per sample because we do not have infinite resources. And so this is a representative set rather than a complete set of everything that could possibly be isolated from these surfaces. So Microbial Tracking 3 in particular is a new campaign to understand the extent to which human-associated microorganisms undergo genetic changes that lead to antibiotic resistance and pathogenicity. So our first objective is completing whole genome sequencing for all of the isolates from Microbial Tracking 1 and 2, and looking at the existence and transmission possibly of antimicrobial resistance and pathogenicity traits that are predicted in those genomes. The second objective is to validate those findings with actual phenotypic assays, so in a C. elegans model or in antibiotic resistance disk assays, and then to do a third campaign sampling the ISS for microbes. And so that's going to be 3 different missions. 2 of them have already taken place. And what makes this particular microbial tracking mission different from the previous ones is that previously we'd only sample once per flight. This time we're sampling for 5 consecutive days in a row. And so this is great because we can actually do statistical comparisons between sampling locations during the same flight. We don't have an N of 1 we don't have an N of 1 anymore for every surface. We have an N of more than 1. And so we can also see how quickly these surfaces on the ISS are reseeded with microbes, or how quickly the bioburden decreases over 5 days of wiping that same surface. And so far we've completed the whole genome sequencing for MT1 and MT2. Those sequences are now all on GenBank and on GeneLab for if you want to access them. And we're also proceeding with whole genome sequencing for all the MT3 strains we find, and for those 424 samples from GSC that Aubrey mentioned, that at least the ones that haven't already been extensively studied and sequenced. So the question of why are we doing whole genome sequencing to this extent? It is truly an enormous amount of sequencing data. What is the use? So one big reason is characterizing the metagenome of the ISS. So anytime you do metagenomic sequencing, a certain percentage of your reads are going to be completely unidentifiable and useless until you know what it is for building models of this microbiome. And so, for example, on the left, you can see an example of where our team found the same unidentified microorganism popping up multiple times in the genome. We're able to go back to the isolates from that same flight, do whole genome sequencing on that isolate, and define a whole portion of this metagenome, and incidentally discover a novel genus Another reason to do it is to track the acquisition or loss of functional genes over time, including predicted antimicrobial resistance and virulence traits. So on the right, you see an example of a strain of Aspergillus fumigatus from the ISS that is more virulent in practice, in phenotype, than clinical Earth strains, but there's nothing in the genome to indicate that. And that, that virulence is something that's retained permanently after its time on the ISS. So we don't really know why. It could be something related to epigenetics, but that's why it's important to be able to compare that whole genome versus the actual phenotype, which is what microbial tracking 3 is all about. So these are the initial, or the taxonomic results from the whole genome sequencing of the isolates of MT1 and MT2. So on the x-axis, you can see the flight. So going from flight 1 to flight 8. And so we do see this change in species composition of the isolates over time. Again, this is representative, not total isolates. You can never get total isolates unless you have many, many dollars. But we do see overlap and a significant change in the microbial population over time, but there's no significant change by location. And then if we look at the fungal isolates from MT1 and MT2, the only difference we see is between the MT1 and the MT2 samples, not change over time. We have a pretty consistent and very happy set of microbes from the Penicillium and Rhodotorula. And this is collapsed, these graphs are collapsed to the genus level, so it doesn't look insane, but I do have the species level graphs if you ever want to look at them. So now we've begun analysis of the AMR traits from those MT1 and MT2 organisms. So here is an example from Acinetobacter pidei, which has been isolated twice on the ISS, so 5 strains from flight 2 and 5 strains from flight 6, or 15 strains from flight 6. And so when you're doing these analyses, it's important to consider the AMR profiles for all of the strains of this same species. So if you look, and I did not include this graph because again, it looks insane. If you look at the AMR predicted traits for all species of Acinetobacter pidei, you'll see these 13 core traits, which are reflected in the AMR profile of those isolates from flight 6. So the flight 6 samples are nothing special. We shouldn't be surprised to see these traits there because any randomly chosen Acinetobacter pidei on Earth would have them. However, the flight 2 strains are very interesting because they're missing some core AMR traits that most Acinetobacter pidei have. And in particular, they have these genes for the ABC efflux pump, which very, very few Acinetobacter pidei strains have. In fact, the only ones that do are some strains isolated from wound and sputum samples at the University of Pittsburgh Hospital. And we also see a aminoglycoside nucleotide transferase, which breaks down streptomycin, which is usually horizontally gene transferred from other organisms besides the Acinetobacter. So this means this is a great jumping off platform for looking for those genes in the metagenome and seeing if this happened on the ISS. And so incidentally, if you're doing whole genome sequencing on an environment to this extent, you're going to get some novel species. So our lab has isolated 14 novel species to date, 5 of which are now in publication. And I'm currently working on Conella hashimotodes, which is a novel Conella species isolated from the panel above the waste hygiene compartment. It's closely related to a lot of plant endophytes, and so we're going to be looking at its application for plant growth. And so we're doing a lot of this work because this is our only built environment in space, and if we don't actually seed these environments with a diverse collection of microbes intentionally, what would we have to work with on a deep space mission if we needed to develop new antibiotics or new applications for biosynthesis? Could we actually do it with the microbes that we already have? Do we actually need to think intentionally about that microbial diversity on these missions? So with that, I'd like to acknowledge all of the organizations that have helped this project, especially our flight implementation team for MT3, and our collaborators at UC San Diego and the astronauts who took our samples. And then I'll take questions, but I'll also do a little plug for Megan and I. We're the two postdocs on this project. So here's little pictures of us and our background before we started. Thank you.
Thanks, Dr. Simpson. And you received many questions, but we'll ask one since we're already over time so people can move on to their next session. Someone would like to know, can you explain what parameters you use to choose which colonies to sequence? Are you purposely choosing different ones or ones that look alike?
For sequencing, the answer right now is we choose all of them, but if you're meaning which do we isolate from the plates, we usually use just difference in morphology. So if like we have like clearly like 10 penicillium and then one thing that's not penicillium, we'll take the one thing that's not penicillium. So again, this is more somewhere between qualitative and quantitative, which most people would call qualitative, but that's the selection process.
Wonderful. Thank you so much. And thank you to all of the early career speakers and for the audience for coming and staying to enjoy this session on space biology.
I just wanted to say, feel free to head on to whatever you need to. We're just going to play that last video that Kevin had. You don't need to stay, but if you're interested, feel free to stay and watch. It's a 5-minute video.
Ignition sequence start. All engines up.
We have
Ignition sequence start. All engines up.
We have taken tremendous steps.
We choose to go to the moon because this decade is out.
We have achieved the earth-shaking, the breathtaking, the groundbreaking,
One small step for man,
and left a mark in the heavens. Our successes build one upon another and amplify what is possible.
The dawn of Orion.
It's time we take the next great leap. We're building the next chapter of American exploration, returning to the moon to stay, so we can go beyond to Mars, to expand what's possible and further our understanding. The architecture for these missions is already taking shape. We will go with new systems, bold designs, and a sustainable mission. You can hear it, taste it, touch it. We are going. We are training, testing, pressing our pioneering spirit into every component, defining our resolve with every line of code, and securing our success with every welcomed partnership. This is not hypothetical. This is not about flags and footprints. This is about sustainable science and feeding forward the advance of the human spirit. Because we are the pioneers, the star sailors, the thinkers, the visionaries, the doers. And because we stand on the shoulders of giants to go farther than humanity has ever been, we will add our names to the rolls of the greatest adventurers in history. Every day, every mission, we advance this call. We are NASA. And after 60 years, we're just getting started.